to continue on to learning objective 3, which is to compare the movement of molecules across the plasma membrane by carriers and through channels. So this is where we really want to emphasize the idea here that passive transport can happen freely through the membrane, or it can require some support. So passive transport is not synonymous with simple diffusion, right? Really important to separate those two ideas. So simple diffusion is one type of passive transport because it's happening down that concentration gradient. But when we move things through a carrier or through a channel, it is also happening down the concentration gradient. So it is still going to be an example of passive transport. This just happens to be through a carrier or through a channel. The first example of this is facilitated diffusion. So the characteristics of a carrier, which will help particles get through the membrane, is that they are transmembrane proteins. And if we think back to the proteins that we looked at on Monday when we described the membrane and some of the different types of proteins that are inserted in that membrane, the carriers are always going to be transmembrane in that they span the full length of the membrane, which makes them integral proteins in that they're always present in the membrane. The second factor here is that they have binding sites for specific particles. So a glucose transporter or glucose carrier is going to have a binding site that is unique to fit glucose. And then the third feature here is that they bind on one side at a time. So if we look at the example at the bottom, we have a glucose transporter. It is inserted in the membrane. Now, by definition, it must span both sides of the phospholipid bilayer, right, in order for it to allow particles like glucose to get all the way through the membrane. So it must be a transmembrane protein and not a one-sided protein. The other thing is that it's going to bind glucose on one side, so it's going to have a unique fit for glucose. It's going to have a conformational change or configurational change, and then it's going to spit glucose out on the opposite side of the membrane. All right, so while it's moving according to the concentration gradient, glucose needs help to get through the membrane. It's too bulky, right? It's not nonpolar or lipid soluble. And so it is going to move down its concentration gradient, but it's going to need these carriers to help it do that. Hence the word facilitated diffusion. If we think about the relationship of the concentration gradient and the net movement for facilitated diffusion, we'll see that the rate of transport of each carrier will determine how much movement takes place, the rate of movement, as well as the number of carriers in the membrane, okay? So whereas with simple diffusion, we saw a directly proportional linear curve, right, a linear relationship, here we have a limiting factor. We need to have transporters to get glucose, for example, through the membrane. Therefore, even if the concentration gradient rises exponentially, the net flux or the net movement is going to be limited by the number of carriers that are in the membrane, as well as the rate of each individual carrier. So the rate of transport of each individual carrier and the number of carriers inserted in that membrane will be the limiting factor. And so we're going to see that the curve is linear to a point, but as soon as we max out those transporters, the curve will now become a straight line, and we're going to no longer see that a higher concentration gradient results in a higher rate of net flux. Okay, because the limiting factor here is the transporters. Now, a classic example here is the GLUT4 transporter. This is what helps to insert or transport glucose across the membrane. And so whenever we need to exert more movement for glucose, we're going to simply upregulate the insertion of GLUT4 transporters into the cell membrane. And because these are proteins, they're going to be synthesized on rough endoplasmic reticulum, which is basically where we synthesize proteins that are made to be inserted in the membrane, right? If you remember this from your foundations, right? If you make a protein on a free ribosome, which is not attached to the ER, then that's going to be a protein that will be exported outside of the cell. But because these are made for the membrane, because they are transmembrane integral proteins, they'll be made on rough endoplasmic reticulum. They're going to be packaged in vesicles, and then they're going to be exported to the membrane, or transported rather to the membrane. They're going to be inserted by stimulation, which occurs from the release of insulin. So because insulin is the hormone that regulates our glucose uptake, whenever we have a glucose-rich meal, if we have a lot of carbohydrates in our meal, we're going to spike our insulin release, and insulin will signal the cell to insert more of these GLUT4 transporters, which will help with that uptake of glucose. And this is how we can move glucose from the blood, where it can be dangerous. This is how we can associate this with diabetes. So if we're not uptaking glucose appropriately, if we're not bringing it into the cell so that it can be utilized for cellular respiration, etc., then it's going to be lingering in the blood. And in the blood, it can be very damaging. It can be damaging in the kidneys, it can be damaging in the eyes, it can be damaging in the peripheral neurons, in the fingers and toes. And so this is where we get to see some of the symptoms that are associated with diabetes, which is where we have low amounts of insulin, whether that be naturally by type 1, right, genetically by type 1, or acquired by diet and lifestyle, which would be type 2. In both of those scenarios, there is decreased insulin, so we cannot upregulate or insert those GLUT4 transporters, and so glucose inappropriately is lingering in the blood, and we begin to see high blood glucose levels or high blood sugar levels, and then that can have damaging consequences in some of those areas, right, of the body. So that is just an example of how we can use our knowledge of facilitated diffusion, which happens through carriers, to really begin to understand some of the clinical symptomology of diabetes and other diseases that behave like diabetes as well. Okay, I think we're just about out of time here. We're going to pick up on Friday with our channels, diffusion through channels.